Hello everyone. In this video, we will talk about chemotherapeutic drugs. The most important concept of chemotherapeutic drugs is antibiotics and its classification. Let's start. Antibiotic is an agent that either kills or inhibits the growth of microorganism. The antibiotic excludes substances that kill bacteria but are not produced by microorganisms such as gastric juice and hydrogen peroxide. Okay, they also exclude synthetic materials, synthetic antibacterial compounds such as sulfonamides. Penicillin is the first antibiotic discovered by Alexander Fleming. Classification of antibiotic. The classification of antibiotic is based on different bases. First of all, on the basis of mode of action, antibiotic are two types, bacteriostatic and bactericidal, while based on the spectrum of action, the antibiotics are broad spectrum and narrow spectrum. Okay, on the basis of mode of action of antibiotics, the antibiotics are of two categories, bacteriostatic and bactericidal. The bactericidal includes penicillin, cephalosporin, fluoroquinolone, which includes ciprofloxacin, ofloxacin, norfloxacin, jamimoxy, levofloxacin, glycopeptide, which include vancomycin, monobacterium, and carbapenems. Okay, while bacteriostatic antibiotics include tetracyclines, actinomycin, sulfonamides, macrolides, chloramphenicol, trimethoprim. Okay, types of antibiotic based on their structural similarity. Some have beta-lactam rings or beta-lactams. Some are macrolides, some are fluoroquinolones, some are tetracyclines and aminoglycosides. The classification of antibiotic on the basis of mode of action. Some antibiotics are inhibitor of DNA synthesis, while some are inhibitor of bacterial protein synthesis. Some are inhibitor of bacterial cell wall synthesis, while some interfere with metabolism and some interfere with nucleic acid metabolism. Okay, in this diagram we can see the targets of antibiotics. Some antibiotics which inhibit the cell wall synthesis include penicillin, cephalosporin, vancomycin. Simply we can say beta-lactam antibiotics, vancomycin. Some antibiotic causes disruption of cell membrane function, polymyxin, while some antibiotics inhibit translation. These include tetracycline, erythromycin, streptomycin, chloramphenicol. Some antibiotics inhibit the folate metabolism. These antibiotics include sulfonamide, while some inhibit transcription. These include rifamycin, which is anti-tuberculotic drug. Some inhibit DNA replication, which include the inhibition of DNA gyrase, DNA topoisomerase type 2 and type 4 by quinolones, fluoroquinolones. Bacterial cell structure. This is not important for pharmacology, but this is important in the understanding of microbiology. So we will not study this bacterial cell structure. Okay, the cell wall, very important protein in the cell wall is porins. These porins is important in the permeability of beta-lactam antibiotics. These porins provide pathway for antibiotics so that they can reach cell membrane and cytosol of bacteria. The cell wall include outer membrane and peptidoglycan, and between the peptidoglycan and cytoplasmic membrane is periplasmic space. This periplasmic space contains the beta-lactamase enzyme. In the cytoplasmic membrane, there are specialized proteins which are called penicillin binding protein. They are also called transpeptidase because they catalyze the transpeptidation, which creates the cross link in the peptidoglycan chain, which is present in the cell wall. While the outer membrane is present in the gram-negative bacteria, this outer membrane is absent in gram-positive bacteria. Structure of peptidoglycan. The peptidoglycan is a carbohydrate composed of alternating unit of N-acetyl nuraminic acid and N-acetyl glucosaminic acid. Okay, NAM polymer. Transpeptidase enzyme. This enzyme is important in the cross-linking reaction. So the transpeptidase is also called penicillin binding protein. The critical part of the transpeptidation is the recognition of dextrorotatory alanine sequence of N-acetyl muramic acid, sorry, not nuraminic acid. Okay, the interfering with this recognition disrupt the nucleic acid, okay, sorry, cell wall synthesis. The beta lactam antibiotic mimic the structure of alanine residue, and they bind to the active site of transpeptidase, which is also called penicillin binding protein, and they disrupt the cross linking process. The transpeptidation mechanism is written here. Okay, the transpeptidase create the cross link between the individual polysaccharide chains. Okay, the penicillin inhibit the transpeptidase, which is also called penicillin binding protein. Coming to the beta lactam drugs, the beta lactam antibiotics contain beta lactam in their molecular structures. We don't have to memorize this molecular structure. Their classification include penicillin and cephalosporin, which are major beta lactam, while other beta lactam include cefamycin, carbapenems, oxacephalosporins, beta lactamase inhibitors, and monolactam. Beta lactam structure is written here. How do they work? The beta lactam antibiotic bind to the penicillin binding protein. The penicillin binding protein is able to cross-link peptidoglycan chain. The bacteria is unable to synthesize a stable cell wall. And finally, the bacteria without cell wall is called spheroblast. Okay, spherocyte, or also called spheroblast. And finally, the bacteria without any cell wall is autolyzed. Mechanism of beta lactam antibiotic. The beta lactam ring has amide group, which is very reactive due to ring strain and conformational arrangement. The beta lactam acetylate the hydroxyl group in the serine residue of penicillin binding protein, and this is irreversible action. Okay, coming to the next slide, mechanism of beta lactam antibiotic. This is not very important mechanism. Next, coming to the natural penicillin. The natural penicillin include penicillin V, also called phenoxymethyl penicillin. It is effective against gram-positive but less effective against gram-negative. It is treatment for streptococcal skin infection, tonsillitis, anthrax, rheumatic fever. The rheumatic fever is caused by Streptococcus species, while anthrax is caused by Bacillus, while tonsillitis is caused by Corynebacterium diphtheriae. Their characteristics are narrow spectrum, should be given orally, and they are susceptible to beta lactamase. Aminopenicillin. Aminopenicillin include ampicillin and amoxicillin. Ampicillin is active against both gram-positive and gram-negative bacteria. It is used for treatment of ear infection, sinusitis, urinary tract infection, and meningitis caused by meningococcus, Haemophilus influenza, and Streptococcus pneumoniae. Their characteristics include broad spectrum, can be given orally and parenterally, and they are susceptible to beta lactamase. Amoxicillin once again is active against gram-positive and gram-negative, and its important side effects include stomatitis, easy fatigue, nausea, vomiting, diarrhea. Coming to the anti-staphylococcal penicillin. The anti-staphylococcal penicillin include methicillin, which is effective against gram-positive bacteria, specifically Staphylococcus aureus. They should be given parenterally, and they causes interstitial nephritis. Coming to the oxacillin, it is also effective against gram-positive bacteria, and it is used for treatment for penicillin-resistant Staphylococcus aureus. This is also very narrow spectrum penicillin and should be given parenterally. It causes hypersensitivity and local reactions. Coming to the nephicillin, the indications, effectiveness, mechanism of action are similar to methicillin and oxacillin. Cloxacillin, it is effective against staphylococci that produce beta lactamase. Very narrow spectrum, should be given orally, and it causes allergic reaction. Decloxacillin, it also causes allergic reaction. Fluoroquinolones, sorry for my bad pronunciation. It is effective against gram-positive bacteria, specifically Staphylococcus aureus, which produce beta lactamase. Next come anti-pseudomonal penicillin, which include piperacillin and ticarcillin, carbapenicillin. Okay, piperacillin, it is active against gram-positive and gram-negative bacteria, extended spectrum, and they should be given by intravenous or intramuscular route. And piperacillin plus tazobactam are beta lactamase inhibitor. Okay, Carpenicillin, it is also effective against gram-negative but has limited gram-positive efficacy. It is used for urinary tract infection, highly soluble in water and acid labile. Its high doses causes bleeding and hypokalemia. Next, ticarcillin. Ticarcillin is very important against Pseudomonas aeruginosa. Its side effects include bleeding, diarrhea, fever, fainting. Next comes cephalosporin. Okay, the classification of cephalosporin is based on their generation system. This is based on chronological sequence of development and also include antibacterial spectrum plus potency. These are bases for their multiple generation system. The first generation cephalosporin are primarily active against gram positive and they have no special activity against gram negative. Okay, these include cephalexin, cefadroxil, cefazolin, and cephalothin. Waiting for loading of slide or some issue of net. Okay, the second generation cephalosporin include cefuroxime and cefotitan. There are many couples of cephalosporins which are second, third, fourth, fifth generation. The third generation includes cefotaxime, ceftriaxone, ceftazidime. The fourth generation includes cefepime, cefepirone, while the fifth generation includes ceftarolone. Carbapenims. The carbapenims are very important groups which include the imipenem. They are a class of beta-lactam antibiotic which has very broad spectrum antibiotic structure. Efficacy. They have structures that provide them highly resistance to beta-lactamase. Carbapenem antibiotics are derived from thinamycin, which is naturally derived product of Streptomyces catalia. The carbapenem common uses the imipenem, meropenem, ertapenem, doripenem. The ertapenem is very broad spectrum, covers gram positive and gram negative, specifically extended spectrum beta lactamase producing strains, Pseudomonas anaerobes. Meropenem, it causes less seizures as compared to imipenem, while ertapenem has no activity against Campylobacter and Pseudomonas. It has limited activity against penicillin-resistant pneumococci. Imipenem. Imipenem is effective against both aerobic and anaerobic bacteria, gram-positive, gram-negative bacteria. Very broad spectrum given by intravenous route, and it is resistant to beta-lactamase. Side effects are seizures. Meropenem. Meropenem is also effective against aerobic, anaerobic, gram-positive, gram-negative. Ultra-broad spectrum. Ertapenem, broad spectrum, gram-positive and gram-negative bacteria. Okay, waiting for loading of another slide. Okay, up till now, please like this video and subscribe our channel. Next class is Monobacterium. The Monobacterium are effective against gram-positive, gram-negative and anaerobic bacteria similar to that of imipenem. Characteristics are broad spectrum, resistant to beta-lactamase, and their side effect includes nausea, vomiting, diarrhea. Beta-lactamase inhibitor resemble beta-lactam antibiotic structure. They bind to beta-lactamase and protect antibiotic from destruction. They include clavulanic acid, sulbactam, tazobactam. Beta-lactam resistance. Okay, the resistance acquired by bacteria against beta-lactamase antibiotics. What is resistance? Drug resistance means unresponsiveness of microorganism to antimicrobial agent. The resistance are of two types, natural and acquired. The natural resistance, which means that some microbes have always been resistant to certain antimicrobial agent. They lack the metabolic process or target site that is affected by particular drug. For example, gram-negative rod are normally unaffected by penicillin G, while Mycobacterium tuberculosis is insensitive to tetracycline. While acquired resistance means that organism was before the resistance was sensitive and after resistance this organism become resistant. The mechanism of resistance include porins, altered penicillin binding protein and beta lactamase. There are many mechanisms of resistance. First is drug inactivation by beta lactamase penicillinase. Second is decreased permeability or change in shape of receptor. Third is activation of drug pump, multi-drug resistant pump, and last is use of alternative metabolic pathway. Challenges of beta lactamase. Okay, we don't have to cover this topic for this slide. Beta lactamase. Beta lactamase enzyme functional classification. It has four groups: cephalosporinase, group 1, group 2, penicillinase and cephalosporinase, group 3, metalloenzymes, and group 4 include penicillinase. The beta-lactamase enzyme, the molecular classification on the basis of serine-based and metallo-beta-lactamase. The serine-based are class A, class C, while B, D are metallo-beta-lactamase. Molecular classification also again serine-based and metallo-beta-lactamase. But in this classification, the serine B or serine based include class D. Extended spectra beta lactamase. Extended spectrum beta lactamase are enzyme that mediate resistance to extended spectrum third generation cephalosporin, which includes ceftazidime, cefotaxime, ceftriaxone, monobacterium, aztreonam, but doesn't affect carbamazine, which includes cefoxitin, cefotitan, or carbapenems, which include meropenem, imipenem. Why should we detect this enzyme? Because the presence of extended spectrum beta lactamase producing organism can result in treatment failure. Okay. Risk factor, we don't have to cover this slight risk factor. Metallo beta lactamase, they are resistant against broad spectrum of beta lactam antibiotics. These include antibiotics of carbapenem family. This class of beta-lactamase is characterized by ability to hydrolyze carbapenems and by its resistance to commercially available beta-lactamase inhibitor. The most common bacteria that make this enzyme are E. coli, Klebsiella pneumonia, Pseudomonas aeruginosa. The beta-lactamase inhibitor resemble antibiotic structure. These include clavulanic acid, sulbactam, tazobactam. Thank you for watching this video and please subscribe our channel for more pharmacology videos. Thanks for watching this video.